Hello and good morning again. So this audio is about our next module. So we're moving on to next module. We finished the DNA recombination module, and today we will be in this lecture we will be talking about transcription. So we'll talk about transcription in prokaryotes first, and then we will move into the transcription in eukaryotes. We will talk about general promoter structures and function, and then I will talk a little bit about E. coli RNA pole structure and function, transcription initiation and elongation. I will not cover termination today, but we will stop there. So we will pick that up in the next lecture. So this is a part of chapter 13th of the molecular biology of gene, Watson and Watson et al. Most of this lecture is from page number 429 to 447 if you want to read more. Okay, so before going into transcription, I just wanted to talk a little bit about the rules of nomenclature, because you all are writing your literature review right now for your literature assignment. So just keep in mind a few things when you're writing about DNA, about a gene name or its RNA or its protein. You will see that the common rule is whenever you're talking about a gene, you will italicize that word. For example, Dunn's in italics means Dunn's gene. And fruit fly, so you won't say Dunn's gene, because once you italicized it, that's what it means. When you're talking about messenger RNA or protein, so if you're talking about the transcript of Dunn's, then you will say Dunn's mRNA or Dunn's transcript. If you're talking about protein, you might say Dunn's is a phosphodiesterase, or which will mean that it's a protein. So, or Dunn's is an enzyme, or Dunn's is a structural protein. So whatever it is, then you will not italicize it. So it's usually redundant to write italicized name of the gene followed by gene. So that's not what we usually do in scientific writing. Okay, so that's all about nomenclature. Another thing that's a lot of students make mistakes with is organism name. So you're writing your genus and species. Your genus starts with a uppercase letter. So for example, you're talking about Drosophila melanogaster. So your Drosophila D will be an uppercase. Melanogaster M will be lowercase. Once you have written your full name of your organism, Drosophila melanogaster, in the rest of your document you will only write D melanogaster, so D dot melanogaster. So we do not usually repeat the genus name over and over. Genus name is just said once, and then you will follow just the first letter of that genus name. Your organism's name will always be italicized. So Drosophila melanogaster or D melanogaster, whatever the way it is, it's always will be italicized. Okay. All right. So we're going to talk about transcription today. So what is transcription? As you all know, transcription is the process of producing RNA from a DNA template. So DNA template that will be transcribed into RNA. That means it's expressing. It's similar to replication in some ways and different in others. So we'll talk about that. The similarity is that the template will be read in 3 to 5 prime direction, and RNA will be synthesized in 5 to 3 prime direction. Like in DNA replication, it's the same way. The DNA is synthesized 5 to 3 prime direction, but it will be read on both templates, right? But when we're talking about transcription, you will be using the 3 to 5 prime DNA molecule as the template to make 5 to 3 prime of the RNA. So here, this is your DNA duplex. This is RNA is transcribing. This is template strand. And this is RNA. RNA and DNA hybrids do not stay for too long. The RNA just comes out of this duplicated DNA. DNA then wound around make this double helix structure behind the RNA. So it will open in front, well back. It will just keep moving, keep making this double helical structure. So we'll talk about this in the next slide in a little bit more detail. Just like DNA replication, transcription also occurs in 3 stages: initiation, elongation, and termination. Initiation, of course, takes the longest time because that's where everything begins. The RNA polymerase has to find the promoter on the DNA and bind to it, bring everything that all need there. And then once the initiation starts, elongation doesn't take long because, again, RNA polymerase is a processive enzyme just like DNA polymerase. And termination will determine by the stop codon on DNA template, and that's where the termination will occur. And we will talk about there are some specific proteins that bind to this termination signal to terminate mRNA transcription. As I said, that there are similarities between DNA replication and transcription, but then there are also differences between replication and transcription. One of the differences is that strand melting is transient in an RNA. In transcription, if you recall from replication, when we talked about that when helicase is in association with DNA polymerase, it opens quite a bit of DNA molecule because it becomes more processive enzyme when it's working with pol. And it opens, the replication bubble is much larger than the strand melting in the transcription bubble during transcription. Strand melting is transient. That means the strand melts, makes the DNA, and the RNA comes out of that DNA-RNA hybrid, and behind that the region that's already transcribed, it will make the double helix structure. The RNA doesn't remain hydrogen bonded to template, and that's what I just mentioned, that it's the DNA-RNA hybrid doesn't stay together. RNA comes out of that double helix of DNA. RNA pol does not need a primer, and you know that because we talked about that in DNA replication as well. For DNA replication, it's RNA pol that makes the RNA primer on DNA molecule, and these primers need to be removed to complete the DNA replication. Because RNA pol can have this ability to de novo synthesize a molecule, it does not require any primer. Okay. So here, this is what we just talked about. This is RNA polymerase. It finds a promoter region on the DNA. This is the upstream DNA. It's always written in positive. This is downstream DNA. This is the plus. This downstream DNA is always written in positive. Upstream is written in negative numbers. The +1 means transcription start site. That's where the transcription will begin. The promoter, the RNA polymerase binds to this promoter. We will talk about this in more detail in the next slide. This is called a closed complex because the DNA hasn't melted yet. The strand hasn't melted. It hasn't opened yet. Once the DNA opened, the double helix opens here. It's called the promoter melting or open complex. This will begin the transcription. That means the NTPs now can come and bind here. This shows the initial transcribing complex where this RNA synthesis had started. Usually 2 RNTPs come first, and those bind here, and then after that the next RNTPs will come. And here you can see that this RNA molecule, as the elongation complex move further, the RNA molecules comes out of this the pol, so it doesn't stay along with the DNA. So as it's moving, this comes along out from the pol, exit channel, and at termination the RNA pol will dissociate from DNA molecule, and here is your RNA. In E. coli RNA polymerase, E. coli has only one RNA polymerase. Eukaryotes have multiple RNA polymerases that we will talk about for the different kind of RNAs. There are different RNA polymerases in eukaryotes. The different subunits of RNA polymerase, just like DNA polymerase. These subunits in E. coli are usually are called the main subunits of E. coli RNA pol are alpha. This is like a scaffold for other subunits that it can bind via carboxy terminal to further upstream regulatory regions. We will talk about that later as well. And then beta and beta prime. These beta forms phosphodiester bond. This binds to NTP. So if you think about our DNA pole, you will remember that right-handed structure of DNA pole. RNA pole is more like a pincher structure. So it's more like just think about the crab's pinches. So it's the beta and beta prime makes those two pinches. Beta prime binds to DNA. This is a zinc finger motif protein. It strongly binds with beta. It's also beta and beta prime work together. This omega is a structural protein or a scaffold that binds to beta prime. So here you can see, so this is what I was talking about. This beta and beta prime make these little pincers, so that will bind to, this will hold the DNA molecule. Let me show you. This is alpha here. This is alpha and beta bind together. This is the N-terminal domain of alpha. This is the C-terminal domain of alpha. The C-terminal domain has because it's bind to this N-terminal domain via a linker. This is a flexible structure. It can bind to upstream regions away from the promoter as well. Okay. So this alpha, beta, and beta prime, they together make the core polymerase. This core polymerase is active during elongation. So remember, we're not talking about holoenzyme yet. This is the core polymerase. So this is the core polymerase, this alpha and this beta prime. The closed fingers, those pincers, they wrap around the DNA strand. Beta and beta prime, they form this site cleft. These contain 2 magnesium for catalysis. One magnesium is bound tightly, other enters with each NTP or RNTP and exit with the pyrophosphate formation. Whenever a base is added, pyrophosphate is released. Remember, because there are 3 phosphates, only 1 phosphate is required for forming the bond. So other 2 phosphates, the pyrophosphates will release, that will produce energy. Sigma is an initiation factor. So this is sigma. Sigma initiation factor has 4 different parts of sigma. So sigma 1, 2, 3, 4. Sigma 2 and 4 are very important. That's what we will be talking more about. This initiation factor promotes tight binding to promoter. So this is the promoter region. And this promoter region that's bound by these sigma molecules. This promotes dissociation from non-promoter DNA. So if it's bound to a wrong promoter or wrong sequence, it will dissociate because the sigma will not bind very tightly to a non-promoter DNA. The sigma initiation factor promotes initial 10 nucleotide polymerization, and we will talk about that just in a minute. This sigma is this alpha and beta, this all together make the holoenzyme. However, the sigma will not stay throughout the elongation. It's only stay there during initiation. So again, if you look at the structure, this is the beta here. This is the beta prime, and then you will see the sigma here. So sigma 1, sigma 2, sigma 3, and sigma 4. The sigma 2 and sigma 4 are very important because these bind to this minus 10 and minus 35 sites of the promoter. So minus 10 and minus 35. So these stay there. They keep the polymerase right there on the promoter tightly in order to start the initiation. This ability to bind DNA, the polymerase core, and promoter dissociation from non-promoter DNA, these functions are provided by different domains of sigma. There are multiple different sigma domains. As I said, there are 4 domains that we just saw in that picture. There are at least 5 different genes that code for these sigma factors in E. coli. Most common gene is sigma 70. There are others that have different molecular weights and different affinity for different promoter structures. So multiple genes can code for this protein. So RNA pole holoenzyme is sigma plus core. So core is your alpha, beta prime. The transcription will initiate close to promoters. So promoter is any sequence that bind RNA pole. So this any sequence is, there's a core promoter, this basal promoter. These core promoters are specific sequences. These are not any sequence, but that sequence that RNA pole recognizes. That's where the transcription site is. That's what your promoter is. These promoters are a type of control elements. These are cis-acting elements that regulate transcription. And when we talk about elements that are cis-acting or trans-acting, that means cis-acting is on the same DNA molecule, where trans can be on a different DNA molecule. So cis-regulatory elements are present on the same molecule of DNA, whereas trans can regulate the gene that are distant from the gene where they are transcribed. So they can be far away. They can be on a different DNA molecules altogether. That's trans. Again, this just shows the holoenzyme. Holoenzyme is together with there's a sigma along with alpha and beta. It makes the holoenzyme. So in E. coli, the promoter recognizes by this sigma 70 shared two characteristic structure. There are two conserved sequences, each 6 nucleotide long. So here these are two 6 nucleotide long sequences. These are separated by a non-specific stretch of about 17 to 19 nucleotides. And these are your -10 and -35-ish. So -10, -35 sequences. These the first transcribed nucleotide is designated as +1. So this is your transcription start site. It's called +1. So all nucleotide downstreams of this +1 are positive. All upstream are negative. So if you're talking about this is -10. This is -35 because these are upstream of transcript start site. Anything if you say +43, that means the 43 nucleotide from the transcription start site. The core promoter has these two elements. This AT-rich -10 box in E. coli is called Pribnow box. And the core promoter element is -35 box. This is the sequence for this is usually TTGACA. This -35 is optional for some promoters, but -10 is required for all promoters. The strong promoters, the highly expressed genes, usually have an up element upstream. So this up element is upstream element. In addition to this -10 and -35 core promoter, this is an upstream element or upstream promoter. This usually provide additional binding site for RNA pole. For example, ribosomal RNA genes have this. And remember that alpha has that CTD structure. That CTD, the CTD, because it's bound to NTD by a linker, so the link is bound here, but then because of the flexibility of linker, the CTD can bind upstream elements. Promoters have consensus sequences. So consensus sequence, as you all know, the most common nucleotide for a given position. So these sequences are not exactly same in all promoters, but there is a consensus. Say, for example, there is a TT, N, CGT, N, CC. So that N can be any nucleotide as long as the others are similar. The sequences of different promoters vary slightly. That's why there's a consensus sequence, because that probably provide better regulation of expression by varying the sequence a little bit. There are 3 stages of transcription: initiation, elongation, and termination, just like replication. Initiation is the rate limiting step. That is the most important level of transcription control. It's similar to DNA replication, where initiation is the rate limiting step that takes most of the time. And once the initiation is over, the elongation doesn't take that long. Initiation requires assembly of the holoenzyme to the promoter. So this is the RNA pole holoenzyme. Holoenzyme means it's along with the sigma subunits as well. So these sigma subunits will bind to promoter, and it will keep the rest of the core enzyme very tightly bound to the promoter. Sigma 70 factor, this can be divided into 4 regions, 1 to 4. Region 2 and 4 recognize -10 and -35 elements of the promoter. There are 4 steps of initiation. Your textbook talks about 3 different steps, but there are 4 steps of initiation that we will go into now. The first step is closed promoter complex formation, which means the holoenzyme, it scans the DNA until it finds the suitable promoter, and then once it finds the suitable promoter, then sigma 4 will bind to -35 box, sigma 2 will bind to -10 box. So this is also very important to remember here, if sigma finds a sequence and it binds there, but then it recognizes that this is not the correct sequence, it will remove from there. That's why it just scans there. So it scans, it stops when it finds a somewhat similar sequence. If it's not similar, it will move on. So sigma will not be tightly bound until it finds this consensus sequence on the promoter. So right now at this step, DNA remains double-stranded. It's a closed complex. So this is we are here. This is RNA pole. This RNA pole scans the DNA. It comes and binds to the DNA, and this is closed complex. This DNA is still double-stranded. DNA hasn't opened yet. This holoenzyme usually cover anywhere from +3 to -44. That includes that -35 site and beyond that -35 site. So this shows here kind of this, if this is -35, this is -44, and it goes further out here as well. So this is, if this is initiation site, if this is +1, it goes up to +3. Then open promoter complex, the next step in transcription, is open transcription. Open promoter complex formation forms when promoter melts. It's here, the promoter region melts, it opens up, the double-stranded DNA helix opens. So helix opens between -10 and +1. There's no ATP required. It's just because the binding of protein to DNA distorts the DNA, and that distortion is enough to provide structural changes in the DNA molecule, and that will open the DNA. So here this opens between -10 to +1. So here, so that's about 12 to 13 nucleotides are now open. This is called transcription bubble. Once a transcription bubble is formed, first two ribonucleotides or RNTPs will brought into the active site. These will align on template strand and join together. You remember most of the time the very first codon is AUG or ATG. So this AUG, so the first two will be A and U that come to the initiation site. The beta will interact with position +3. It's a local single-stranded DNA, and it makes this open promoter. If there is an up element, then the CTD, the C-terminal domain of alpha subunit, will bind there. Because CTD is bound to NTD, it will stay there. Okay, another point that I wanted to make here is at this point, once those two nucleotides are added, once it's open complex, the polymerization will occur even if it's not, even if the transcription abort afterwards. But the transcription will add these two nucleotides an additional 9 nucleotide. Okay. The third step is polymerization. In polymerization, up to 9 nucleotides are transcribed while the holoenzyme remains stationary at the promoter. What that means is the holoenzyme, the sigma subunits, they're still bound to -10 and -35 at this point. They haven't moved away from the promoter. The polymerase hasn't moved from the initiation site yet. It's still staying there. So what it will do is it will pull the DNA inside the polymerase. It's called scrunching while it still remains on the promoter. And then it will add these 9 nucleotides. So first nucleotide is usually an A, and after that whatever will come is usually AUG to make methionine as first amino acid, and then whatever the sequence determines will be the sequence of the transcript. Last step, the fourth step, is promoter clearing. Once if these 10 nucleotides are transcribed, the first nucleotide plus 9 more are transcribed, then RNA displaces sigma from exit channel. So the sigma will move because it weakens the affinity of sigma for RNA pole. Sigma dissociates from the core. The core enzyme now moves down to the template strand, and this is elongation. So here, this is bound. This sigma element is bound to -10 and -35. Keeps the holoenzyme here at the promoter region. It will first add the 2 nucleotides, then another 8 nucleotide or up to 9 nucleotides will be added, and once it's add those nucleotides while still staying here, this is the transcription has now begun. It goes into elongation. This is RNA exit channel. So you will see that polymerase has different exit channels. So these channels will determine the exit of these sigma subunits. So sigma will move. Once the sigma moves from here, then the pole can move over around the DNA molecule. Second stage of transcription is we talked about initiation. RNA pole comes and bind to the promoter region of DNA, and it will add about 10 nucleotides during initiation. Then it goes into elongation. During elongation, the synthesis occurs in 5 to 3 prime direction. It will require the reading template in 3 to 5 direction, and about RNA polymerase adds about 50 nucleotide per second. This elongation requires topoisomerase activity, so it's very likely that DNA is wound in front of RNA, and it needs to be negatively supercoiled. Elongation involves this transient transcription bubble, so it's a small portion of DNA will be opened at a given time. RNA will be transcribed there. Then it will move. The pole will move next. Will open another small portion of DNA to make this transcription bubble. This is about 8 to 9 nucleotide of RNA within the bubble form a heteroduplex with template, and that's pretty much it. Only these 8 or 9 nucleotide of DNA and RNA stay together. The rest will come out. It's called the processive strand synthesis, which means elongation continues until specific termination signals are encountered, and those specific termination signals are wherever the stop codon is in DNA. So the transcription is processive because the RNA pole is adding multiple nucleotides at a given it's in a single catalysis reaction. The strands enter RNA pole as double-stranded DNA. So this is the double-stranded, as we talked about in the beginning, the closed DNA complex. It's still not open. So double-stranded DNA enters into RNA pole. Then it's separated, moved through different channels. There are total 5 channels here. So some of these you can see in this example. There's NT channel, T channel. There are total of these 5 channels. RNA exit channel. So this RNA pole has these different channels for different processes that's going on. The template strand passes through the active site. It's briefly hybridized to the RNA product, and only 8 to 9 nucleotides stay hybridized, and then it will come out, move out. The non-template strand of the DNA, that will be the 5 to 3 prime of the DNA, will be out. We shunted out that single-stranded DNA. It will not enter into the polymerase RNA pole. RNTPs these enter through the active site through their own channel for polymerization. So they have, this is the NT channel. That's where these will enter into the RNA pole. The single-stranded DNA re-anneal behind the polymerase. So this is your single-stranded DNA, double-stranded DNA. This if it's working right here, this will re-anneal. Proofreading. RNA pole has the capability of proofreading, so it won't make many errors. However, you will realize, or if you think about it, proofreading is a whole lot more important for DNA replication than mRNA and then the transcription. Although it is important for transcription because you don't want to make a wrong sequence to leading to a wrong protein. But if the proofreading is not foolproof in replication, then that has a capability, that's a possibility of mutating the genome itself, the organism itself. While if it happens in RNA, it could be just for that one time, that one protein, because the gene sequence is still intact. So proofreading is still important, but probably not as important as for replication. There are two basic types of proofreading mechanisms for RNA polymerase or for transcription. Pyrophosphorylated editing, that means when there is a wrong NTP is entered, RNA pol will pause, it will reform, it will break that NTP by breaking the phosphodiester bond and replacing it with the recently released pyrophosphate. So that the recently released pyrophosphate will provide the energy for this editing. Hydrolytic editing is another type of editing mechanism. In hydrolytic editing, the 3 to 5' exonuclease activity, which is very similar to DNA pol, but it's stimulated by different factors called GRE factors. These GRE factors will stimulate the RNA pol to start 3 to 5' exonuclease activity. This will enhance the hydrolytic editing, and it will also act as an elongation stimulating factor. So these GRE factors, they act as elongation stimulating factors, so they are helpful, or they're required for this elongation of RNA polymerase. At the same time, they're also checking, they also have this exonuclease activity. Another group of proteins are called NUS proteins. These NUS proteins join polymerase in the elongation phase, and these promote the elongation and termination. These are more important in termination, and we will talk about these proteins when we're talking about termination. There are another proteins called TRCF. This is a helicase. It's a transcription repair coupling factor helicase. So if RNA pol stalls, that means it stops for any particular reason. It can be due to a wrong NTP has entered, or it could be because there is a mutation in the gene there. So these TRCF helicase, these stay on double-stranded RNA behind RNA pol. If the RNA pol stalls, it will catches up to RNA pol, and either it will push the RNA pol past the block, whatever the problem is, it will just let it go, or it will make it dissociate. So it's depending upon the problem, the issue. It might just help it get through that problem, or it just, if the problem is too big, then it will make the RNA pol dissociates. Then TRCF can perform both polymerase removal function and repair enzyme recruitment. So if it's only if the problem is too big and the RNA pol has to dissociate, then it will bring the repair enzymes, and those repair enzymes will repair the DNA molecule before transcription can begin. Termination. It depends on terminator sequences. These are classified as, there are two different type of termination sequences, row dependent or row independent. Both of these mechanisms require hairpin formation, but I will stop here today. We will talk about termination in our next lecture.